Hi, everyone. Thank you so much for joining our presentation today titled High-Throughput, Low-Cost Transcriptome Profiling with the Applied Biosystems Clariom GO Screen Assay. It's my pleasure to introduce you to Anthony Schweitzer. Anthony is the Principal Scientist and Director of Bioinformatics for Expression Microarrays at Thermo Fisher Scientific. For Anthony's complete biography, please visit the tab at the top of your screen. And if there are any questions during this presentation, we encourage you to submit those in the Q&A box. Anthony will address your questions following the presentation. Please join me in welcoming Anthony Schweitzer. I'll now turn the presentation over to him.
Thank you, Maggie. Welcome, everybody. Thank you for joining us today. Today, we're going to talk about RNA gene expression or transcriptomics, but specifically today, we're going to talk about a high-throughput, low-cost system we call GO Screen. And what's really great about this system or platform is that the all-in price point per sample is $50 or 50 euros, depending on what location you're at. But basically, it allows you to do thousands and thousands of samples, reading out a transcriptome profile. There's very flexible sample input. You can use either purified RNA, or this has actually been designed to work with direct lysis, which you'll see is quite convenient for people who are doing screening hit profiling. This application is currently only available through partnerships with two different service providers that we've negotiated with. So this is not run inside your lab. However, you would send samples to the service provider and they would run it for you. Now, just to orient you as to how this platform compares to other microarray solutions that we offer, if you look at this slide over here on the far left, we have our discovery solution, which is the Clariom D solution. The D stands for deep, and this allows you to get a very deep view of the transcriptome. This is a microarray that can read over 6 million different probes. You can look across every well-annotated gene in our transcriptome and many unannotated genes or content we call speculative. The resolution of this particular microarray is exon resolution. So it's like casting a very wide net to do discovery types of work, especially if you're looking for biomarkers. When we move into more profiling and screening, people tend to focus on well-annotated genes, things that you would find in the RefSeq database or the Hugo Pathways database. And for these applications, we have the Clariom S, as well as the product we're going to talk to you today about, which is this GO Screen product. Now, these assays essentially will look at well-annotated genes and give you essentially a pathway level analysis of what's happening. If you would look in the literature, what people publish on, most people publish on well-annotated genes, essentially. It's only if people really want to do either discovery that they would go and use something like Clariom D versus what we're talking about today, which is more profiling. Of course, to continue the RNA solutions that we offer, there's also verification solutions. And these are panels, essentially, that people would use for routine clinical testing research, things like TACMan and OpenArray, and any of the AmpliSeq panels that exist out there for Thermo Fisher. Now, the product that we're talking about today, this GO Screen product, is actually a 384 peg plate that you can see down here being pointed to by this red arrow. On the human version of this particular peg plate, we can fit what is essentially 20,000 human genes. And within that 20,000 human genes, you get essentially all of the genes in the gene ontology database, everything in Wikipedia Pathway, any of the biomarker associated genes that are out there. If you're used to looking at different panels by other platforms, things like immunology markers or inflammation markers, they all exist already in this collection of 20,000 genes. So again, you don't have to sacrifice what you're looking at when you can look at 20,000 different gene expression events using this particular platform. Of course, we keep other content on here as well. We have housekeeping genes, gender-linked genes, which are useful for doing quality control of samples. And in this human GO Screen plate, we actually have also included some human viral content. Now, that content is often used to actually set up and develop a screening application within a compound pharma screening that would be done by the pharmaceutical companies. It's conceivable that you could measure viral content with this, say if you're screening human samples. However, that's not the intent of this content. This content was actually there to monitor the viral load used in the screen itself. Now, much like human, we've also developed a version of this array on rat, and for that we have the Clariom GO Screen Rat. And here we have 21,597 rat genes. And again, much like the human version or human counterpart of this, we focused on the well-annotated genes for rats. Now, one interesting thing about rat is that most of the companies out there are using one of two different versions of the rat species here. We've done here empirical probe selection with samples using the Han-Wistar rat, as well as the Sprague-Dawley assembled genome. And this was important because if you go to your databases and map essentially probes and develop gene models, that is currently all being done with the Sprague-Dawley. And what was important for us to do is to make sure that any SNPs or genetic variation in the Han-Wistar would be accounted for in this particular design. And so we were very careful to do that. Basically, if you're doing any kind of rat models and screening with them, this is the array that you would want to use. Now, the GO in Clariom GO Screen is actually referring to the gene ontology resource. And this is a very important database for many of our pharmaceutical partners. They essentially are able to break out the gene content of our transcriptomes and use this resource to get information in terms of the cellular components, the biological processes, and the molecular functions of the genes that we've represented on these particular microarrays. Now, the application that is most used today for these GO Screen products is pharma secondary compound library screening, which is essentially hit profiling. So what happens is these pharmaceutical companies are taking these vast databases of compounds and they're profiling them on various cellular assays, and they get in the order of tens of thousands of hits, basically, from these various compounds. It's then important to get more information on these hits, and they use systems like GO Screen to essentially understand the profile of the gene expression that is actually coming from that particular screen. Now, other customers have been interested in using these arrays for other applications, such as studying immunology and inflammatory disease. And we have customers who are actually taking patient-derived cells, they're sorting the cells, and then they're profiling these cells in cellular assays, again, using different compounds, but a much smaller number of compounds. And these types of studies can basically help identify responders and non-responders, as well as patients who are undergoing some type of adverse effect. And it allows you to kind of go in and study, more importantly, what's happening in these particular immunology inflammation cell types. If you think of 12 different individuals who might be studied here, you might sort those 12 patients out into 12 different sorted cells, and you might then take each different sorted cell and profile it with different compounds. And so these experiments can explode up into large numbers very, very quickly, and that's why this high throughput system works very well. The rat product that we have out is actually being looked at in terms of using it for toxicology, specifically mechanistic toxicology. And you can imagine that looking at these gene expression profiles can actually help you quickly identify the toxicity mechanisms that are happening in certain types of events within these animals that are being tested. We've had people also contact us about ag bioscreening and yeast model screening applications as well. Again, the idea here is we're working with tens of thousands of samples and can provide up to 20,000 profiles at a very low cost. Now, using gene expression profiles to classify and stratify compounds is actually something that has been validated and understood for quite some time. Here's a paper that was actually developed by a group from Bayer Scientific. David Rourke is one of the authors on here. But they were one of the first people to actually show that you can actually train a system like this to develop a signature or profile for these various compounds. And that allows you then to essentially predict the response you might get or the profile you might get from new or novel compounds. And this was quite valuable to be able to actually do this in some type of screening as opposed to trying to develop this and test in animals or humans, obviously. So one of the main things we've had to do to develop this system is to fit essentially 20,000 genes on a very tiny peg that we can put into our high throughput system. The way we went about this was we started with our older systems and looked at our Clariom S and Clariom D platforms. Here, typically, we're looking at around 10 probes per gene. And what you see here on the right is essentially a gene model. There's many different transcript isoforms that can be spliced in different ways. And you can create essentially a gene model looking at all these different exons of this particular gene. What we typically do is we would put down the 10 best probes in various exons across this gene model to get essentially a summary of the gene expression event that was happening here for this gene. Now, to get a smaller chip, basically, and to reduce the cost, what we've done here is we've focused the content on the best 3 probes. And we've done this by doing both probe-based selection using in silico ranking and empirical probe selection using real data that we've accumulated over the years on these various microarrays. Notice that we're also trying to target probes in the most constitutive regions here of the gene. So basically, we're trying to get a gene level estimate as opposed to trying to understand the splicing and the differences of the exons here. When we go and we look at the performance in a classical Latin square study using spikes, we can compare this performance essentially to our other products. What you see here is our Clariom S gene level product compared to the human GO Screen products, at both using all 10 probes and using the 3 probes that we've selected. And essentially, we get the same performance in terms of sensitivity and specificity. Now, these plots that you're looking at here, these are called receiver operator characteristic curves. And again, what this is telling us is that we can actually look at a very low level event here. This is actually looking at 1.5 copies per cell versus 3 copies per cell on average in an RNA sample. Okay, and so this is looking at a very low differential expression change. And what you see here on the right panel are those spikes, and you see a blue distribution of the spikes. And this is the T statistic or t-test saying, yes, I see it differentially expressed, versus everything else on the microarray, which is the null distribution and should not be changing. That's all of the other genes that aren't spikes. This is a very nice way for us to clearly look at and gauge essentially the performance of these microarrays. So we've picked essentially the best 3 probes, and we achieve a performance level that we're satisfied with for these types of applications. Now, if you ask, how does this compare to other technologies like RNA sequencing? Well, we can actually take the same RNA samples that were used during the Microarray Quality Control Consortium and were essentially applied to both TaqMan and RNA sequencing. And we can show that the fold change correlations are essentially equivalent to what we get with these other technologies using purified RNA. So again, we expect these arrays to work much like these other technologies in being able to find differentially expressed genes. Now, the Clariom GO Screen arrays also contain a lot of controls. We have the ability to look at the general signal metrics to make sure we're getting out good signal from the genes we expect to see expressed. We also have controls for the hybridization. This is both the array and the scanning controls. And then we have target prep controls, which actually make sure that the RNA was in fact purified and or amplified correctly. In addition to that, we have these very nice positive negative controls. These are actually exon intron controls, and we expect to get good signal from the exons, obviously, and no signal from the introns. If we start to see signal from the introns, that means we're amplifying DNA and not RNA, and that would be problematic. So this is a nice set of controls that we also have on the array. We have what we call anti-genomic controls as well. These are probes that are used to estimate signal background and can be removed. Help to remove the background from your probes during the pipeline processing that happens. As I mentioned earlier, we also have the viral content on here for the human genes, And we've included the ERCC controls that were made popular during the Microarray Quality Control Consortium. The good news is you don't really have to worry about these controls because your service provider will look at these controls and use them to make sure that the assay is working correctly. And more importantly, you can actually analyze this data in our software, which is Transcriptome Analysis Console, or we call this TAC. We've designed this for investigators, although it's very sophisticated and a lot of bioinformatics people do use this. If you're not familiar with this, it's one of the biggest advancements we've had in our array portfolio. It's free. It works with our new and older arrays. And basically, it allows you to retain that ownership of your data so that you can actually analyze it and get your results and move on much quicker with your research. There's already over 600 literature citations where they've published papers essentially using this software and using the figures that come out of this software. And down below here, you can see some very nice figures. We have the principal component analysis, the differential expression analysis, and the ability to do things like hierarchical clustering. If you're not familiar with this, with some of our other products, you can also do microRNA association analysis as well, and pathway analysis. Now, when we're working specifically with GO Screen, what we're really trying to do is we're trying to understand what the transcriptome profile is for a particular compound in a particular cell type. And what I'm showing you here is actually some real data. And then in the center, the large PCA plot that you see here, there's actually 6 different cell types that have essentially been applied with different compounds. And each one of these cell types, essentially, you can see kind of breaks off into its separate cluster, meaning that that transcriptome profile is somewhat similar within a particular cell line. But if you then focus your analysis within each cell line, you can actually see that different compounds at different concentrations will actually give you a differential expression or a different transcriptome profile. And this is what basically creates the signature, essentially, that somebody can then use to evaluate a new test compound compared to standards or known compounds that have previously been run. And if you look at the top left panel here, you can see very clearly, if you focus on this DMSO control, it's actually the blue that's over here in the left cluster along with 2 other compounds. And so the DMSO is the vehicle control to get the compounds essentially into the cells. So in 2 of the compounds here, we're not getting much of a transcriptome response here, but you can see these other 3 compounds are actually giving us a very unique transcriptome profile characteristic of those compounds. And this is quite a powerful way to essentially look at this data. Now, again, the beautiful thing about GO Screen is that we're looking at 20,000 gene measurements. And previous technologies, if you're thinking about using things like BioSpider, I think they use about 3,500 different gene expression profiles. Or if you look at the L1000 technology, the Luminex bead technology, that's only looking at 1,000 genes. Here we're able to actually focus a transcriptome profile on 20,000 genes. And this is really the power of this particular platform. One thing I also want to introduce you to is something called dimensional reduction algorithms. Here's an example of some data using something we call EGA, or exploratory grouping analysis. And this comes in the TAC software. What's nice about these dimensional reduction algorithms is that it goes and it looks for clusters of transcriptome profiles, essentially. So it helps you build, essentially, this structure of your data and allows you to then find similarities based on other things you know about the compounds or other responses that you've collected over the course of your history doing any type of screening or clinical trials. This particular tool we're using is called TSNE. TSNE is quite popular these days. It's used for all kinds of things, for looking at patterns in traffic, as well as internet usage patterns and things like that. So these are quite powerful algorithms. And when you can take 20,000 pieces of information for every compound and throw at these particular algorithms, it can be quite powerful in finding associations. Just to be clear, when we use these different algorithms or incorporate them in our software, we always have an advanced parameter mode. So if you're a bioinformatics person out there, these tools, although they're quite easy for a normal person to use, you can also get quite sophisticated in your analysis by using the advanced parameters that we've also made available. So originally, a lot of this work was done using something called the connectivity map, or CMAP. And it's a really brilliant resource that was originally constructed by the Broad, which uses transcriptional expression data to basically look at all of these relationships between diseases, cell physiology, and different therapeutics. And these signatures essentially are sitting there and can be used today to essentially go and profile compounds. There's over 100 different cell types in the database, and there's over 27,000 different compound concentration combinations that are in there. Now, this database was only generated really across 1,000 genes, and these were landmark genes that were originally actually found on microarrays and thought to be the best representation of genes. So basically, to get the cost down, they used this Luminex bead technology to profile 1,000 genes here. And what's been found is that this 1,000 genes was probably selected, tailored more for cancer than for these other types of screens. And so the thought is what would happen if we actually go and use 20,000 genes as opposed to just 1,000 genes. However, this data resource still exists out there, and people still want to leverage this vast resource. And so if we want to, we can actually go and try to compare now GO Screen data to CMAP and the profiles that are in CMAP. Now, to be able to do that, you want to use what's called the level 5 CMAP data. And what I'm showing you here is that you can actually take the differences between the compounds and the vehicle control in these screens. And these gene expression profile differences are actually very similar between the two platforms. And so the triangles here actually represent GO Screen. And for this particular compound that's highlighted here, it's kind of the salmon pink color here. This is compound AT7519. You can see that these profiles are all clustered together here. And within this profile, there's different cell types, which is why you see the separation that you see here. Now, there is some technical variance you can see in the different platforms. However, what's good here is that you can still leverage and get some estimation of what your compound looks like relative to the data that exists in CMAP. In fact, you can actually build a prediction based on an enrichment scoring technique. And when we did this with these 6 compounds across these 5 different cell types that you see at the top here, it turns out we could actually correctly predict, in most cases, the top prediction was both the correct cell type and drug in 16 out of the 34 cases using this enrichment score algorithm, essentially. Now, in some cases, that wasn't our top hit was not the actual compound. However, it was often within the first 2 or 3 compounds that we would assay, essentially, in terms of the hits. And then you have these other cases where, in fact, you see the same mechanism for a different compound that is essentially known to have the same effect, essentially, by the same mechanism, which essentially translates down to the same pathway. And that's what we see here in this case with the persimetab here. Now, if we really want to talk about screening, it's really important to understand that we've enabled this particular technology to work with direct lysates. And this is new for microarrays. Typically, we only work with purified RNA. We've designed this platform to work with something called cells to signal, which is a commercially available direct lysis solution, basically. It's already been tested and works with a number of different cell lines. Most of our testing on this particular platform was actually done with HeLa and HepG2 lysates. And to understand how this works in your lab, typically when you're running these screens, you're working with 96-well plates or even smaller plates, or in some cases larger plates. But the idea here is that you want to directly lyse these cells. And to get a good lysate that works with our assay, you want to make sure that you do an ice cold PBS wash on those cells. And then what you want to do is you want to basically target a concentration of 100 cells per microliter using this lysis buffer. Different screens will have different cells, and the cells themselves will be of different sizes. What we will typically do is we'll work with the customer to ensure that this lysis process is happening efficiently with that customer. But again, the two main important things here are you want to make sure you do this very cold PBS wash with the samples to remove all the media, and then you want to target your lysis solution on the cells at about 100 cells per microliter. When you go and you actually send your samples to the service provider, you're going to be sending them probably in a 96 well plate. If you're already working in 384 well plates, you can do that as well. But we do request that you leave a couple of wells blank so that we can add some HeLa and liver RNA controls. And these are batch processing controls that allow us to QC essentially what's happening at the service provider. And when we talk about actually working with a partner such as yourself, typically we go through these different phases. There's a pilot phase. Here's again where we like to determine that the cell line is actually working with the lysis under the right conditions. In some cases, we might actually do a second dilution in the lysis buffer. And we might also adjust the amplification cycles that we're actually using should we be working with some very different type of cell. This allows us to ensure basically that everything is working well up front. Then typically there'll be a small POP study where the customer will run known compounds with known effects so that we can basically demonstrate the effectiveness of these transcriptome profiles. And we do all of this, again, ahead of the full screen. And again, these screens can be quite large. We're talking in order of 10 to 40,000 samples in a screen. And that allows us essentially to get down to these very low cost price points. Now we've partnered essentially this service offering with IBX here in the United States. This is formerly known as Rutgers, or this was the Rutgers service provider. They've recently changed their name to Infinity Biologics IBX, however, and they are in New Jersey. So in the United States, this is your primary service provider. And if you're listening to this recording from Europe, we've partnered with Eurofins, formerly known as Aero Supplied Biotechnology out of Denmark. Now, both of these groups we've worked with quite extensively, and we really value their participation in this platform because they've been really great partners and generate really high quality data. What typically a customer will do is they'll actually order this lysis buffer, and then they will work with us and send essentially their samples to these service providers who will then buy the arrays and the chips and run the actual service. So with that, I will thank you for your attention and take any questions.
Thank you so much, Anthony, for that outstanding presentation. We're going to go ahead and move into the live Q&A, but we are running short on time, so we'll probably only have time for about one question. So the first one I have here is, when compared to the alternative screening platforms like RNA-seq, why is GoScreen the best choice for research?
Maggie, thanks. That's a fairly common question we get, and the answer is twofold. One is cost, and quite simply, even though the cost of bulk RNA sequencing has come down quite a bit, the cost of doing library construction, especially thinking about doing library construction across tens of thousands of samples, is quite prohibitive. The other thing is that we're still working with RNA, which means you need dynamic range. You need to make sure that these signals or these profiles that you get, if you're measuring gene X in one transcriptome profile, it might be represented 10,000 times, where gene Y might be there 5 times. And then in another sample, gene X and gene Y might be there 5 times for both genes. And so the number of reads you need to see those genes can change quite a bit. And so you need a minimum depth, essentially, to see those gene expression profiles. And when you put all of that together across 20,000 genes and try to do it with sequencing, that depth becomes quite prohibitive. Now, if you try to target it with panels, first of all, it's quite difficult to put together panels of that size, of 20,000. But more importantly, across these large screens, if you're building databases across time, it becomes prohibitive because these panels that you build have variation to them in their manufacturing. And so you'll have dropouts and things like that that happen. What's nice about microarrays is that semiconductor technology creates essentially the same microarray every time. So it's really great for these studies that go on for long periods of time that basically are building databases. So it allows you to directly measure all of these 20,000 events on every single chip. You don't have to worry about dropout.
Thanks so much, Anthony. And thank you to everyone in the audience for the great questions today. As we have run out of time, just a reminder, any questions you did submit that were not answered will be addressed via email. And don't miss out on all of our other valuable presentations as part of this virtual event. You can visit the Agenda tab in the auditorium for a full listing. To receive more information about the new Clariom GO Screen Assay, please visit our microarray analysis booth in the exhibit hall, the poster hall to see GO Screen posters, or click on the red Request More Information button throughout the virtual event. Lastly, this presentation will be available for on-demand viewing through September 30th, 2021. Please share it with a colleague who might be interested. Thank you all again for your participation. Until next time, goodbye.